Good day, ladies and gentlemen. My name is Rastin Jade RJ Dr. Baramo, and we are group number 1. We are here today as a group to demonstrate how to characterize bacterial culture on agar plates, agar slants, and nutrient broth. Without further ado, let's get started. But first, here are the group members. We have Miss Alfonso Chelsea, Miss Basal Lirame, Miss Bautista Frechette Cristela, Miss Beramo Rastin Jade RJ, and Miss Bonus Ana Pamela. Our agenda for today is to enable us students to distinguish basic features of bacterial culture on agar plates, agar slants, and nutrient broth through a demonstration video. Secondly, to enable the students to recognize the advantages and limitations of cultural characteristics in the identification of bacterial species. Now, before we proceed with the discussion, let us first define cultural characteristic, or commonly known as colonial morphology of bacteria. It is one of the most fundamental aspects applied in the identification of bacteria. This describes various features of bacterial growth on a medium such as shape, edge, elevation, color, texture, and many more. Moving on, let us not wait any longer. For our first speaker, we have Miss Bautista Frechette Cristela to present the characterization of bacterial culture in agar plates. Miss Bautista, you now have the virtual stage.
First characteristic we have is pigmentation or color of bacterial colonies. Pigments in bacteria play important roles in the survival of the organisms which produce them. Among microbes, bacteria is mostly capable of producing pigment with different varieties of colors, including Chromobacterium violaceum, which gives off a purple color, Cycocyanin, which gives off a green color, Astaxanthin gives a pink-red color, and the carotenoids, which are organic pigments produced by plants and algae, and they usually vary from yellow to red pigments. The second characteristic, which is the surface characteristics, bacterial colonies are frequently shiny and smooth in appearance. Other surface descriptions might be dry or powdery, wrinkled or rugose, mucoid, contoured, concentric, and lastly, radiate. For the third characteristic, we have shape of the colony. There are 3 points given in identifying the form or shape of the colony. This includes size, surface, and color. Some examples are punctiform, circular, filamentous, irregular, rhizoid, and lastly, spindle. The fourth characteristic is size of colonies, preferably in millimeters. First one is pinpoint, pinhead, and matchhead. Now, for the fifth characteristic, we have margin of the colonies. These are entire, undulate, erase or serrate, filamentous, curled, and lobate. For the sixth characteristic, we have the elevation of colonies. These are flat, raised, umbonate, convex, effuse, and subsurface. The second to the last characteristic is odor. Some bacteria resemble a variety of odors, which can be a pungent or sweet scent. Bacteria can grow almost everywhere and produce directly the odors themselves. For the last one, imparting pigments to surrounding mediums. One example of this is the McConkey agar.
After we tackle characterization of bacterial culture in agar plates, let's talk about naman sa characterization of bacterial culture in agar slants. Before we proceed, we must know what is agar slants. Agar slant is a culture made of slanting surface of a solidified medium in a test tube that has been tilted to provide greater area for growth, and also can be used to culture bacterial cells for identification. So we already knew the agar slant. Let's proceed to examination of agar slant culture. First, the amount of growth. We have 4 categories to identify the amount of growth. On a single line streak on agar slant, we can identify whether growth can be scanty or slight, moderate, abundant, or no growth at all. After identifying the amount of growth, we can move on to the color of the colony. As we observe in the picture provided, while the bacteria grow in agar slants, some bacteria are chromogenic, means they grow in different colors. Some may grow in yellow, green, blue, or black. Some chromogenic bacteria were proposed as an etiological factor in the production of black pigment. Periodontal pathogens such as Porphyromonas gingivalis. We can also do the optical characteristics, means we can identify. So, what are the 4 consistency of the agar slant? First, it can be butyrus. Butyrus is a butter-like texture. Second, it may be viscid, with growth follows needle when withdrawn. And the third one is membranous. It is a thin, coherent, or like a membrane. And lastly, the brittle or dry. We can include also in the optical consistency characteristics the growth, the transparency of the slant on a single streak. The outcome may be opaque, not permitting the light to pass through, or translucent, that can some light passes through but not enough to permit visibility of the objects. We can encounter also opalescent. It is like an opal color. Or the iridescent. This is like exhibiting changing rainbow colors when the light is reflected. And sometimes it can be fluorescent, just only having one color. We can also identify here whether it is dull or not glossy, or glistening, the glossy texture. And lastly, they may form and characterize as filiform, confluent colonies all throughout the line of inoculation. Second thing to characterize was the growth extendiform along the line. Third is the echinulate, toothed or pointed margin, and we can also encounter bedded, the separation or confluent colonies at about the half of inoculation. And lastly, the rhizoid. They are irregular, branch or root-like in appearance, or some are branch-like, tree-like growth. And lastly, effuse.
Now that we have an idea on the different characteristics of bacterial growth in agar plates and agar slants, it is time for a new media. representing our nutritional broth. Before I talk through the characteristics that can be observed on bacterial growth in our nutrient broth, let's refreshen ourselves with what is nutrient broth and how do we make it. Of course, the quick and simple way. So, what really is a nutrient broth and what is it for? Nutrient broth is a liquid culture made out of meat or vegetables, but most likely meat. It is a liquid culture media, and just like nutrient agar, we can also mix blood, body fluids, serums, and etc. And with that, it could accommodate fastidious or bacteria that are high maintenance. Now, what do we need and how do we make it? Let me tell you, this is going to be quick and easy. All you need is distilled water and nutrient broth powder. Yes, you heard me right, just those two. Now, what do we do with those, you ask? Well, we just need to combine the distilled water, and which could be an Erlenmeyer flask or a conical flask, and finally sterilize it through autoclaving. This step is very, very important, so please do not forget. Okay, let's say that we already have our baby bacteria culture in our broth. What do we expect? Well, there are 5 things to observe in our bacterial culture in nutrient broth. Mainly the surface, the subsurface, the amount of growth, the sediments, and the pigmentation. Even though no is always an option for these guys, we still need to make ourselves aware because this will help us distinguish and narrow down our long list of possible bacterial cultures present in our broth. So let us start with the surface of our media. We are to expect 5 outcomes, which are clear, membranous, pellicle, ring, or flocculent. Let's start with the left side. First on the left, we have flocculent. So flocculent is a type of surface buildup that consists of numerous small, irregularly shaped growth. This looks like a bunch of small islands floating near but not touching one another. Next, the ring. And just like its name, the growth forms a ring or a buildup on the edge or near the wall. Next is pellicle, and this is a continuous unbroken biofilm, and it usually appears thicker. And last but not least, we have membranous, which is a thin growth just like a membrane. The next characteristics are really easy to identify because they are pretty much straightforward. Starting off with the subsurface growth, and yet again, we have 5 possible outcomes. We have clear, turbid, granular, flocculent, or flaky. Well, clear is pretty much self-explanatory. Turbid is when the broth is murky or it so appears to be blurred. Granular is the presence of small granule-like growth, and flocculent is just like the surface growth, small, scattered, irregularly shaped growth. And last but not least, we have the flaky, which is pretty much similar with granular but make it bigger. Next characteristic we need to observe is the amount of growth. This time we only have 4 possible outcomes: none or no growth at all, scanty, which means small amounts, moderate with more growth, at least more than scanty, and of course abundant, which literally means a lot, like a lot. Next, we have sediments. We have granular, which have small granules, flocculent, with small irregularly shaped masses, flaky, with flakes, and last, viscid, with coherent or uniform swirl. Okay, as for the final, we have pigmentation. This time we have a lot of possible outcomes, and it actually depends on what kind of bacteria we have. Then again, it could be clear or colorless, but then it could also be not. The bacteria could produce pigmentation of sorts like pink, red, yellow, purple, and many more. But please be advised that not all bacteria can produce their own pigmentation.
When it comes to assessing agar slime culture, one of the most important factors that we look for is the amount of growth that the organism takes in, as well as the form that it takes. None. If there is no growth in the culture, it could be because of a variety of factors, such as the failure to cool the needle properly or the inoculating loop. Repeating the experiment might cause the culture to fail, and this can also affect the results. A scanty or small amount of growth is usually indicated by an organism during its culture period. Moderate is usually indicated by an organism having neither too much nor too little growth. Finally, abundant. It is usually indicated by an abundant quantity. The development of growth is characterized by the presence of filiform, confluent, or beaded colonies throughout the inoculation. Papillate, on the other hand, is similar to filiform but has a different shape. Villus, on the other hand, is a root-like structure that appears in the appearance of a villi. Finally, arborescent is the branching tree-like growth. The liquefaction of some bacteria occurs when they cause a layer of gelatin to get covered in a liquid. This causes the agar to turn into a liquid. In the process, the bacteria create different patterns on the agar. These patterns are referred to as Characterization, napiform, infundibule, saccate, and stratiform. Characterization, since the surface area was transformed into a saucer shape, it was classified as a Characterization. Napiform, it was distinguished by a bulbous-shaped liquefaction at its surface. Infundibulum is a funnel shape. Saccate, elongated tubular in form. Lastly, stratiform. It was characterized by total liquefaction of the medium's upper half.
And that brings us to the end of our presentation. We would like to take this opportunity to thank the audience for your time and attention. Once again, we are group number 1 from 𝑑𝑉M3C. Thank you very much and have a nice day ahead.